All right, so welcome again. Um, this is the Universal Sequencing TELL-Seq story series that we are doing this time. We have the fortunate and honor to have Dr. Victor Martinez to give us this topic with the short story long, using linked long-read sequencing in clinical laboratories. And the brief agenda today, with this open remark, we are going through the keynote speech, and then we will have a brief Q&A afterwards. So let's give a brief introduction of our speaker, Dr. Victor Martinez. He is the genomic scientist at the IWK Health Center in Halifax, Canada, specializing in clinical genomics, and he is also assistant professor at the Dalhousie University. With a PhD in biomedical science and a postdoctoral fellowship in cancer genomics, Dr. Victor Martinez is also a director of the IWK Center for Clinical Bioinformatics, CC Bio, and leads the data-driven initiatives for precision medicine and the IWK. During his time, he has developed and implemented genome-wide bioinformatic analysis frameworks to pinpoint crucial genetic variants in hereditary cancer and rare diseases. Additionally, Dr. Martinez is spearheading the project to assess the implementation of linked-read, which is TELL-Seq, in the clinical settings at IWK, with the goal of utilizing long-range sequencing information to decipher clinically relevant variants in duplicated regions of the genome. Dr. Martinez's research and clinical work are supported by the grants from Genomic Canada, the Canadian Cancer Society, and the Beatrice Hunter Cancer Research Institute. Among others, this support is also instrumental in his commitment to nurturing the next generation of scientists at the IWK Healthcare Center and Dalhousie University. Now, without further ado, let's welcome Dr. Victor Martinez. Stop sharing here.
Oh, thank you very much. And let me start sharing. Oh, I need to wait until. Okay, so I'm sharing my screen now.
Good, we see it. Thank
perfect.
you.
Yeah, okay. So welcome everyone, and thank you very much for the invitation to share our project about the implementation of linked reads in a clinical laboratory. As mentioned, I'm Victor. I'm a clinical genomics specialist at the WK, and I hope you enjoyed our journey with linked reads. So for this seminar, I'm basically hoping to provide information so we can have a better understanding of the linked read sequencing technologies. I'm going also to discuss some of the methodology and critical steps that we found during the development of this project, and I will show some use cases that I hope can exemplify how linked reads can work in a clinical environment. So the presentation is structured in 4 main modules. I will briefly talk about the WK and the clinical genomics laboratory in our hospital. Then some context, probably you're very familiar with this, but just to provide context between short, long, and linked, or everything in between reads. Then some aspect of the methodology of TELL-Seq, describing the protocol, describing a little bit of the technology. And finally, I will end with some examples of how we can use this for different clinical situations for detecting variants in complex regions of the genome. So starting with the WK, we are a tertiary care hospital in Halifax, Nova Scotia, and we serve women. It's a women's hospital to children and families, especially. It's not only the patients, there's also some whatever we do in terms of genetic diagnostic, it also affects families, so we also take care of that. And our catchment area is the maritime provinces in Canada. So as a reference for those are not familiar with the maritime provinces, I wasn't actually. So it's Nova Scotia, New Brunswick, and Prince Edward Island. And so we are the only pediatric hospital east of Montreal in Canada. So we receive samples and we take care of patients from all this region in Canada. So the Clinical Genomics Laboratory at the WUK is responsible for providing genetic testing for all this geographic area in Canada. We do about 7,500 molecular tests per year. Of those, 1,500 to 2,000 probably at this point are based on next generation sequencing for different conditions. So we through different projects supported by other institutions in Canada, we now are able to provide whole exome sequencing, not for all the patients at this point, but very soon we'll be able to provide whole exome sequencing for all patients as a backbone. And then on top of that, we overlay virtual panels. We have around 40 NGS panels for cancer risk, pediatric adult disorders, and rare conditions too. But importantly, we will collect all this data from patients that will hopefully help to provide better interpretations, since we'll have a baseline of the structure of the genetic structure of the population. We have recently been accredited by Accreditation Canada Diagnostic. That was a huge milestone that we were pursuing for several years. So we just got it earlier this year. And this is on the clinical aspect. In the research component of our laboratory, we are part of different nationwide initiatives supported by Genome Canada. In particular, the All for One initiative, that it's different hospitals, different institutions sharing the vision that if our patients can benefit for their genetic testing and our data, other patients across Canada can also do it. And so we are working together to establish all the data sharing protocols and the infrastructure needed to share this data across Canada and provide better and better care to our patients. So I'll go briefly with the context where linked reads are located in the next generation sequencing or third generation sequencing space. So for short reads, you are probably very familiar with this, is typically 150 base pairs length. So it's widely adopted in clinical environments in Canada, probably worldwide, but in particular in Canada. So Illumina this is not a secret or advertisement. It's a dominant technology in the clinical space, and mostly because this technology has been validated, it's accurate. The certain lines of sequencer have a high throughput that enables to multiplex patients, so we can run multiple patients in a single sequencing run. Locally here on one single run, we can do 24, maybe 32 exomes with an X-Seq 2000 instrument, which is something that it's relatively standard for clinical labs. But also, it is important to know that the bioinformatic pipelines are very mature and well established, and not only from the coding perspective or the robustness of the software. They are very well integrated with the sequencer, and then how automatically data can be transferred and analyzed for a secondary analysis, and then tertiary analysis too. So if we consider a clinical lab, this streamlining all the process is very efficient for our purposes. However, it's not perfect. There are some difficulties in sequencing, and particularly in the repetitive regions of the genome. So what is the main problem that we see in these regions? Is that the performance of short read next generation sequencing is relatively poor. And we are talking about regions where the sequence of a gene is duplicated, for example, in pseudogenes, or if we have repeat expansions, or if any sequence of interest has more than the regular copy, extra copy in the genome. So also with repeat expansions, other genetic events that basically it's the addition of more and more repeats, which make the whole stretch highly repetitive. Those are areas or events that are very difficult, if not impossible in some cases, to detect through NGS. So the problem for duplication is this: if we have a unique DNA sequence where the segments are not duplicated, then we perform sequencing here, and when they need to go to the bioinformatic processes, reconstruction is not a problem, everyone is happy, and then we can continue with our analysis. However, if we get duplicated regions of the genome, like those two red regions here, then the reads that are coming from that particular segment, when we get to the remapping to the human genome, those reads can go to one segment here or the other, sometimes with the same probability, which decreases the quality. And we start with a lot of questions about, I mean, is this the actual position of this read or not? So why is this important? So why is this a problem for next generation for short reads? Well, because there's a high proportion of duplicated sequences in our genome. Could be like satellite DNA, segmental duplications, pseudogenes. But on those regions, we still can find some clinically relevant genes. So this is a not comprehensive list of genes that are probably the worst nightmare for people who works in the bioinformatics field trying to remap. Sorry. There's a question or? So these genes are clinically relevant, and some of them share a high degree of homology with other regions in the genome or within the gene itself. There is different type of homology, could be within the same gene, with other non-functional copies in the case of pseudogenes, or with other non-coding regions in the genome. But the point is there are duplication, so the reads, the short reads, are not able to map correctly to these regions. Now, long reads, it's a at this point not emerging, but it's a very different technology. So it's not 150 base pair anymore. We are going from 1,000 to million of base pairs. So there's no problem at all with duplications. You're familiar probably with PacBio and Oxford Nanopore, two different technologies, two different approaches, but both of them can provide long reads. And the main advantage is you can actually sequence the whole genome, regardless of the degree of repetition. And in fact, only a couple of years ago, thanks to these technologies, we were able to finally know the full sequence of the human genome, thanks to long reads. However, limitations in this case is the error rates of long reads are they're getting better and better, but still higher than compared to short reads. They're high cost, capital cost, because we need new protocols, we need new equipments. So it's something that it's not readily available at this point. And the bioinformatics pipelines, the design for these technologies are still pretty good, but the integration with a fully operational clinical workflow, it's still a little bit challenging. So long reads are a great technology and can solve all the problems that I mentioned. However, for the limitation that I mentioned, it's still not ready, in our case at least, for a full clinical adoption. There are some projects in United States about implementation of long reads, and it's something that eventually will come to the clinic. But at this point, we want to benefit from the long-range information that can be provided from long reads, but also hopefully using the same instrumentation that we have available in our clinical laboratories. And here's what link reads actually can help to bridge that gap between short and long reads. I don't know for how long and the final scope, but it's a technology that is promising in terms of filling this space, so we can at least experience a portion of the benefits of long reads. So linked reads are 150 base pairs in length. Is the same length as a short read sequencing. However, there is long-range information that can be obtained from this region, and that can be achieved because of barcodes that are added to each read, and I will show some more specific examples. But these barcodes are useful to assign a read to a specific DNA molecule, the original DNA molecule, so we can remap to the specific location. And also, it doesn't require new investment because the sequencing protocol for this technology basically uses the same NGS protocol and reagents that you are using at this point. So basically, the main feature for linked reads is using these barcodes that are added during the protocol, the linked read protocol, can be used to reconstruct the long DNA molecule and also put it back to the original location in the DNA. So I know it's a complicated concept in some cases, so I will try to show it with an example. And I had to recognize this example came from one of my students, Marv, that is working very intensively on this project. Is the analogy of a book page. So the book page you can see here, assume is a DNA molecule with the complete the context and all the letters and phrases. So a single phrase in this book will be equivalent to a short read, and the book page number is the barcode. So this is an astronomy book, so the phrase "the sun and the earth" will be fairly common across this book, right? So we're going to find the same phrase, but probably and most likely in different pages. So I know, although it's the same read, I can accurately find the one that I'm looking on page 75, and then using that barcode, put it back into the correct page. However, each one of these words here can be a phrase, and some of them will overlap to each other. So other reads, and remember this is a process that occurs in parallel, will have other reads or other phrases that are also with the same barcode on page 75, and then when we remap, we can put it together. So at this point, we have what is called a linked read, and then we can keep doing more and more. So basically the length of the read, although it's not physically long, like in the long read technology, it's linked to each other and can provide us the context that we are trying to look for long-range DNA. So thank you, Mark, for that analogy. I know if he's probably doing rotation at the hospital, but that was a perfect way to explain how linked reads works. So I'm going to focus a little bit more on TELL-Seq at this point, and their ability to identify or to position reads and provide long-range information on complicated regions of the genome. So the TELL-Seq technology, and just as a highlight at this point, I will show more specific in the next slide. So requires high molecular weight DNA according to the protocol. But I will show you some data, and actually it's pretty new data, that it might work with old samples too, which is, personally, I think is a huge step forward for a clinical implementation. It also relies in barcoding. So the technology uses this barcode to reconstruct the DNA fragment. So it's attached to each read. Reads with the same barcode will be linked together and start reconstructing this book page or the whole phrase within a book. Uses the same sequencing technology. You can sequence on any Illumina sequencer or short read sequencer with the same reagents. After the TELL-Seq protocol, you proceed as any sample. You can actually use the sample in a regular way without considering the barcoding, because this part, the barcodes are useful during the bioinformatic assembly of these reads, which occurs after the sequencing. You will have an extra FASTQ file for those familiar with the bioinformatic process, which will contain the barcodes for this process. So this is roughly how it works, and not too much detail, but I just want to mention the key elements for this technology. So we have the barcoded bits here, our DNA of interest, and hopefully, again, the high molecular weight, the longer the better for the DNA fragments that we are trying to explore, and also the transpososome as a element for the protocol. Then the complex that it's formed between the transpososome, the DNA, and the barcoded gets assembled, and on top of these beads, and then a second transpososome will trigger the tagmentation reaction, where basically the DNA segments will contain the different barcodes that are necessary for the downstream analysis. So this is the structure of a TELL-Seq read, which is pretty similar to a regular Illumina read, with the exception of this 18-base tag that you can actually identify on the FASTQ file, but this is what will be used eventually in the downstream bioinformatic analysis to regenerate the DNA molecule. So now I will switch to some examples, some results of how the TELL-Seq can be useful in a clinical context. And I will focus some of the examples in PMS2, and I will mention other genes too. But PMS2 is just because it was it's probably it's the best example of a gene that is highly relevant from the clinical perspective, and it's due to its function, but it's also very complicated to identify variants that are present in this gene due to homology with other pseudogenes. So PMS2 is an essential component of the repair machinery in our cells, and defects in this gene or pathogenic genetic variants are related to Lynch syndrome, the constitutional mismatch repair deficiency syndrome, also cancer syndrome, and also in hereditary breast and ovarian cancer. So this gene is present in several of our NGS panels. And the problem is that whenever we see a variant that is potentially pathogenic in PMS2, we don't know if this variant is located in the PMS2 gene or on any of the 15 pseudogenes that resemble the sequence of PMS2. So of those 15 pseudogenes, 14 of them cover exons 1 through 5. But there's one which is particularly problematic, PMS2CL, that takes basically the second half of the gene. So in some cases, you can apply some techniques to identify if a variant is located in PMS2 or not. But most of the time, the degree of homology, and particularly when you get to this portion here, like the 3' end of the gene, it makes the variant interpretation very problematic. So in most of the cases, if the molecular cause of a condition, for example, like hereditary breast and ovarian cancer, is located in PMS2, it will remain undiagnosed because we don't have the technology to identify. Some labs in Canada were providing the service, and you can actually get you can get better results with nested PCR techniques that involve around 60 PCR reactions, which is very labor intensive. And even with that, you are not 100% sure that the variant is located in PMS2. So at this point, the only solution that we have for this type of cases is to refer out that particular sample to a commercial laboratory, and at that point we lose the data. We don't have the data back, the raw data, and it also is an added cost. In this case, for the system in Nova Scotia, the patient does not pay for the genetic analysis, but for the healthcare system, it's an extra cost that it's involved in the same patient, which was already sequenced in our facilities. So having long reads and having the possibility to identify variants in prevalence too is, of course, with the first thing you will think, oh, I have an idea. Let's use long reads to detect variants in PMS2, like actually the physically long reads. Dalhousie University across the street of the hospital has a PacBio sequencer, so let's start putting some numbers on something like that. I probably don't need to say much, but this is how my conversation with the hospital leadership went. There's no funds, and from the lab perspective, it's very complicated to change. And for an operational lab, it's very complicated to change the protocols, the equipment, and everything. So that's not going to happen in the near future. We are not going to have long read sequencers in a clinical environment, and then retrain the personnel probably will require to pause providing services and dedicate a portion of the team to get to familiarize with the new technology. So what do you do when there is no clinical validation opportunities? Well, we presented a research project, and the research basically was trying to identify the feasibility of using this technology, particularly TELL-Seq. And I had to recognize that the whole idea started when 10x launched the link read technology. However, you probably know that there were some intellectual property issues. So at that point is, okay, we will need just to wait. And fortunately, the TELL-Seq opportunity appeared. So we decided to test that technology. The other question was, can we identify variants in duplicated regions of the genome confidently? And finally, if we can do it, can we phase the variants? Can we determine haplotype for each one of these variants, which is something that, I mean, we can barely identify the variants. If we can phase them, that will be another huge step forward. And at this point, I want to just mention and thank the Beatrice Hunter Cancer Research Institute for their support and trust, because it's without their support through an early investigator that were, all this work was completely impossible to perform. Also, well, thank to UST for the support on the protocols and bioinformatic analysis, and Illumina because they also contributed in our sequencing endeavors for most of the samples involved in this project. So I'm gonna mention now some of the TELL-Seq highlights, the methodology highlights, and at this point I need to recognize that all of this work, the wet work, has been performed by Marv as a med student that is spending any free time, which is not too much, being a med student at Dalhousie. In this project, he's presenting results in different conferences at this point. So hopefully trying to show to other labs that this is something that it's worth to at least consider. But something that was really important for this project in particular is that Mark had previous experience as a clinical genetic technologist in our lab. So he was familiar with the next generation sequencing protocol that we use, but he was also in a position to evaluate how easy or hard will be to the implementation of this technology, and which points of the protocol will be problematic or maybe that will require some modifications for the clinical implementation. So I'm not going to go through the protocol. You can find all the documentation on the UST website. However, just a few learned lessons from our work with this project. So high molecular weight DNA is important. So handling the DNA, the way that we handle DNA is particularly important. So there's a recommendation of cutting the tips of the pipettes, and this this gentle handling of the DNA also makes the protocol a little bit longer, and until you get enough experience to with that. But again, you can still have data from previously extracted sample, and I will show you what we did in that area. You please use the cleanup procedure that is established in the protocol. We learned that the hard weight, our first batch, we did it. I think it's the recommendation is, I mean, you can do 1 or 2. Okay, let's go with 1. That was our actually regular protocol. However, the first batch of samples that we sent for sequencing, only one of them, one of our 8 passed the QC. Because there were some contaminations. So after we discussed with the UST tech team, we were able to fix that problem. And for the second batch, basically only 1 sample failed. So that was a big recovery. A little bit of delay, but we learned that was important. For the same reason, although the protocol can be performed in 1 day, between the handling of the sample and the cleaning procedures, it's probably better to consider that you will spend 2 days in this protocol. And also importantly, there are 3 safe stopping points, and you can probably see here notes. You can stop at in some step for 2 weeks, for 1 month, or for 6 months, which is important to kind of organize the schedule for when these samples can be sequenced, and if you need to collect more samples to multiplex them in a run, for example. So the bioinformatics portion of this data, it's basically it has 3 different algorithm or steps that you can perform. The TELL-Read, it takes care of the VCL files from the sequencer, converting it into FASTQ, and also generated a QC report. Also, the TELL-Sort analysis will generate all the variants and the phasing of this the different calls that are performed. And also there is a third component, which is TELL-Link, that where you can perform de novo assembly of the data, considering the barcodes. There is also a possibility to, and this is very useful, and a parenthesis here. So the data analysis from TELL-Seq is not automated like a regular next generation sequencing data. It's more similar, if some of you are familiar with Expansion Hunter, for example, where you run the algorithm, you have the data, but still you need to perform some manual curation of the data to get the sample. So you still need to look at the raw data and see what it's telling you about a particular case. So in that context, having all the files on IGV, it's pretty useful. And we, through another grant that we got from ACENET, we were able to implement this portion in high performance computing clusters from the Digital Research Alliance of Canada. This work is being performed by another of my students, Melis, that is a Master of Computational Biology and Bioinformatics at Dalhousie. So we are basically trying to build the whole pipeline and test it with our data, which in this case it's a lot of data. So we want to provide this basically end product for future clinical implementation. So after you run the bioinformatic pipeline, this is one of the main quality parameters that you will need to assess. So this is the super long fragments that the metric that is generated. In the case of human genomes, you want fragments larger than 30 kilobase pairs, and from freshly extracted DNA, all of them look good. So it's kind of the optimal condition. These were our first tests after we solved the cleaning problem. So we were pretty happy with the results. However, this is an ideal condition, but it doesn't necessarily reflect what's happening in the lab. So usually what happens, at least in our lab, is we get the sample, we extract the DNA with another non-high molecular weight kit. Then we process the sample, and at that point, probably after 3-4 weeks, we get a result, and then, oh, we should actually look in at PMS2 or any other duplicated sequence before, depending on those results. So at that point, it's not possible to re-extract that sample. Probably the blood sample is not even available to do it, or it's already degraded. So we had a question is, can we obtain some data or some useful data from samples that were previously extracted? So this is what we did. We used the high-pass plus gels from Blue Pippin to select DNA fragments 30 kilobases or more. It adds 15 hours because you need to elute and pass the DNA, but we were able to obtain kind of a high molecular weight still DNA, not pure, but from previously extracted samples. And now the same metrics, although it's not as good as the freshly extracted DNA, it's still pretty good from a clinical perspective, because in this range, you still have a lot of genes involved. But even if you are investigating one single gene, let's say some somewhat recessive monogenic condition, the whole gene, and trust me that it's much more than one single gene, will be contained in the same fragment, so you can perform the same phasing and analysis for it. So this is the quality portion, but after working with all the TELL-Seq, TELL-Read, TELL-Read and TELL-Sort, the different algorithms from the bioinformatic process, this is what you get from the analysis, and this is the IGV format. So it's a lot, there's a lot of information. So let me walk through the different tracks that you will find as a result. So the first one here is what it's called a phased block. So a phased block is a sector, a region of the DNA where the variants were, the reads were linked and the variants can be phased together. So within the same block, I can tell that variant is in haplotype 1, the other one is in haplotype 2, they are in trans, one is coming from the paternal allele, the other one from the maternal. You can do that within the same block. You cannot do it block to block. So each block basically defines the haplotype number differently, but the haplotypes are, the phase blocks are long enough so you can identify variants within the same gene for sure. And this is actually a PMS2 case. The second portion of the IGV, data that you can see is what's called a pseudo BAM file. So each one of these black lines that you can see here represent an haplotype, and there's a small perpendicular lines that represent variants detected on that particular haplotype. The numbers in between, it's the distance between variants that were identified, and for at least for the variants that were phased during the experiments, and also depending on the sample. Not all variants can be phased, but most of them, and at least you can have some information. The third track here that you can see is a typical BCF file. You have your reference allele, you have your alternative allele, and then at the bottom of that, in this gray-blue line, you have the genotype that is suggested by the analysis. So that's basically a guide to say this is a heterozygous or a homozygous variant for this particular region. And then after that, you can see the typical pileup of a BAM file. So on top is what you typically see from an NGX experiments with the variants highlighted there. But then this track over here is what the reads that were assigned to haplotype number 1, in this case, haplotype number 2, and reads that were not phased because the sample quality or any other phenomenon in this case. So although you cannot phase or use all your reads, there are still some information available in this case. So I mentioned that the PMS2 gene has particularly problematic regions. So I'm going to show you one of them. This is exon 14 and 13, which is in the middle of those problematic regions. So On any regular case, you probably can see the variant. You are not able to see or even to estimate if they are in cis or trans from a regular short read sequencing. So in this case, for example, if you focus on those 3 variants over there, we can see that the support for those 3 variants, all of them correspond to haplotype number 2. So those 3 variants are in cis, and then depending on the condition that you're evaluating, you can assign likely benign or benign, because they are basically not in the opposite allele. You can see, for example, other variants here that are in the opposite allele. So you can start reconstructing, and depending on the phenotype and other conditions, you can see, or it will help the interpretation of your variant. So that's an example with PMS2. I will show you examples with other genes that are also problematic, but basically to put an example that it's not only for PMS2, you can actually do on any genome, on any gene on the genome, or any region that might require this type of technology. So I'm gonna show you a couple of examples with a region that is particularly bad, the CYP21A2 TNXV, and also for cystic fibrosis in this case, how you can identify variants in CFTR, in the CFTR gene. So CYP21A2 and TNXV are related to complicated conditions that will require phasing of this variant. However, for anyone that is familiar with variant interpretation, if you see variants on any of these condition, in any of these regions, it's very complicated to see it. They are very close to what it's called the NGS dead regions. The whole region is flagged as a low quality due to homology. So usually there's very low chances to provide a confident result for anything detected in this region. However, if you have run the samples through the TELL-Seq technology, this whole region here is contained in the same phase block, meaning that we can phase variants in this particular gene region. So, for example, if we move to the 3' exons of CYP21A2 that are relatively, and actually very complicated to identify, so we can see that particular variants in this case, for example, can be phased. So this 3 variants are in cis to each other, and also we know that this variant here is in trans. Although, I mean, even those 2 variants are pretty close in like linearly in the gene, but they are located in different alleles. So that's very relevant information when you're performing variant interpretation, because we can discard some regions, some variants, and focus on the ones that are more likely to explain the phenotype of a patient. The case of CFTR is the same, and in this case, it's a cystic fibrosis. Basically, we're always looking for where is the second variant, where is the second allele affected. So that is not easy to find. Parents are not always available to perform a trio, for example, which is the other way that you can phase some of the variant. So for those cases where there's not both parents are available for sequencing, we can also use the technology. Again, CFTR, the whole gene is contained in a phase block, and actually the phase block goes way over these regions here. And if we start looking at the particular sequence of the gene, each one of these variants and are basically, it's possible to phase them and to provide information to the patient and also to the family. In this case, it's very relevant. Like different from the other conditions, this has implications for the patient in most of the cases, but also for the family, for family planning, and for other type of decisions that we'll make. So in this case, for example, each one of these variants here can be phased. We know that all these variants are in the same haplotypes, so we can start structuring the variant interpretation process. So those are the examples, and now just to end the presentation, I will just try to summarize with some conclusions. So the TELL-Seq technology is a add-on to the routine NGS resources that we have available, and it can improve the diagnostic yield, particularly when we consider conditions that are that can reside in genes where there's some sort of duplication, or also even if there's no duplication, without the presence of parents for testing, we also have the ability to face the variant and identify what is the pathogenic pair of variants that might be affecting a particular, for example, autosomal recessive condition. The integration into a clinical lab, although we still, there's a lot of work to do, but what we think in our discussions is it's a familiar data. So we are not talking about different formats. There is a different bioinformatics pipeline that is required, but once it's implemented, you can feed data to this pipeline and get the data from it. You can use the same equipment, you can use the same NGS reagents, so there's no additional. So it's basically a new test that you can offer. And in terms of time added, it's not it doesn't extend the time, but since it can be considered a new test, the turnaround time for that patient, once you find something in PMS2 that requires further investigation, that basically triggers a new test, which could be the use of linked reads for detecting those variants. So we need to validate this, and in terms of best practices, basically. So we played a little bit with the freshly extracted versus previously extracted samples, the optimal cleaning steps that we need to perform, and other considerations that will be needed in order to be an established test in a clinical lab. But I think we have accumulated enough data to convince some of our clinicians and technologists in our lab that this is something that can provide answer for patients that basically remain the genetic cause of their condition. It's still undiagnosed. And then we're also looking for collaboration with other clinical labs that would like to implement or test the technology, or research institution that would like to test this. Most of our samples were sequenced at Genome Quebec, and we keep having conversation with them. So do you have similar projects? Is any other people working? Oh yeah, we just had a request from other labs. So they are also interested on looking on this technology. Genome Quebec itself was interested on establishing a more robust pipeline, because the data at that point looks a little bit different, but with discussions with the clients, with us, basically, they're starting to understand what is the capacity of this technology. So the next steps that we are envisioning for this technology, first, education and training. This is part of the, so it's not a magic bullet for any duplicated region. It will fail in some samples, it will work perfectly in others, but we need to kind of set the expectations internally and externally for how useful will be this particular technology. And I'm talking about the clinical application, because I mean we are basically in charge of patient care and health. So it's particularly important to communicate what's possible and what's not possible, same as what's possible with NGS or any other technology. We have a kind of more high-risk, high-reward project, and see if they could actually work with repeat expansions, and that requires more probably a de novo assembly of some reads. We are we have another project in the lab where we are investigating the use of NGS for repeat expansions. However, it is limited to the 150 base pair length for short reads. There's no way to, there's a few ways, but usually the most sensitive results will be within that range. But if we can expand that a little bit, we think that we can probably add not only for duplications in a sequence itself, but also to kind of measure the level of expansion occurring in particular areas of the genome. That's another project, and hopefully, I know maybe next year or whenever we're done, we can do another webinar with those results. And finally, I understand that there is some enrichment protocols that might also help to facilitate implementation in this clinical space. So now, just to finish, I want to thank Marv for the huge amount of work he's done in this project, and now he's enjoying a little bit the results and showing and discussing with other people. Melis will be working on the implementation of the pipeline in our HPC clusters, and working with this data to see how easy or what type of other resources we need to bring for bioinformatics in this case. Other people, Karen Bedard, which is part of this project too from the beginning, the whole personnel from the clinical genomics laboratory who allow the intrusion of research people. I mean, familiar, but still we were working kind of these crazy projects that luckily at this point it's showing some results. People from UST, Peter from AceNet, Ross, that was developing the whole, helping us to develop the pipeline in the cluster from Compute Canada, and Genome Quebec for the sequencing of the samples. And again, just to end up with a big thanks to the Beatrice Hunter Cancer Research Institute for the support, and UST and Illumina for the continuous support on this on different aspects of the project. Thank you very much. I'm very happy to answer any questions that you might have now. I'm very happy to answer emails or any other communication that you will like or you will have at this moment. Thank you very much.
Thank you so much, Victor. And then for those who are online, feel free to unmute, and then just first self-identify who you are, where you are working with, and then you can ask the questions. We have about 50 or 10 more minutes. For those that you can also type your question in the chat if you want me to read it out and queue it up. Otherwise, we would just welcome you to unmute and start getting back to the questions for Dr. Tennis. It was a fabulous presentation. Thank you so much. I learned a lot in terms of the phasing.
Yeah, that's something that we actually are. We were very interested, because I mean that's a no limitation, and it's not a criticism to the short read NGS. It's a limitation of the technology, but in our case, we see that continuously. That I mean, sometimes we are in the edge of classifying something as a VUS, because we don't know the phase of the variant. But if we knew the phase of the variant, we were able to say that variant is likely pathogenic, and that triggers a completely different patient management protocol, basically.
All right. There is a question comes up from Geoff Woodland, and said, was this done as a whole genome sequencing or with an exome capture kit?
No, it was done as a whole genome sequencing, but we are very interested to test more an enrichment. I was discussing with DMARC about the new approaches. So and I think that will be, I will be really interested to see data from the enrichment kits, but this particular data was whole genome.
Yeah, just as for the you mentioned that one of the advantage of TELL-Seq is and NGS is to do multiple samples at the same run to save cost. I know that you are using the high throughput kit for 24 samples, so you can run 24 at the same time. We also have the high throughput kit for 96 samples, and now we are building 384 samples, for the index samples primer. So that would also enable more samples to be run at the same time to save a lot of sequencing cost.
Oh yeah, for those with a larger capacity sequencer, those are it's very good actually. I mean, if you have the enough volume of samples, but it's something that it's yeah, will be pretty interesting.
Yeah. Okay, so let me see if there's any more questions. Feel free to mute or type that into the chat. Then we can have Dr. Martinez to answer it right away online.
I see another one, barcode hopping. We control that in the same runs, and we don't see any difference from the I mean from the regular analysis that is performed with we use base and Dragon for analysis of that portion, but yeah, that's something that we could actually explore to something that is more specific to the barcodes and see kind of some possible contamination with other like across barcodes.
Great. All right. So we have but in the interest of time, I would just thank everybody for participating in this webinar, and again thank Dr. Martinez Victor for giving such a fantastic talk. So if you have any questions regarding to TELL-Seq in terms of getting the download of all the publications, or getting any inquiry regarding to how to use the TELL-Seq kit and learned about how to download the software that we have freely available for you, or collaboration with any other labs that we have. And definitely feel free to email me or send, for any questions regarding to TELL-Seq or any questions regarding to our user cases. We are going to have this webinar to upload to YouTube, so everybody can review it at your free time, as well as share the wealth of the knowledge and spread the word. So this applications of the TELL-Seq for the link long read in the clinical lab could be widely adopted and used and benefit for the healthcare and make us more healthier and happier. So with this, I will conclude this webinar, and let me just stop the recording.